Let's begin by acknowledging that diet is not just about calories, weight loss, or macros. Diet is information. Every bite we take is sending signals to our body, to our cells, our immune system, our microbiome, our hormones. And over time, that information translates into health or disease, vitality or inflammation. So when we talk about plant-based versus meat diets, we're not just talking about food preferences. We're talking about biological messaging. And the real question is what kind of signals do these diets send, and what are the downstream effects on our health? Let's start with plant-based diets. Plant-based eating is often associated with a high intake of fruits, vegetables, legumes, whole grains, nuts, and seeds. These foods are rich in phytonutrients, natural compounds like flavonoids, carotenoids, and polyphenols that have powerful antioxidant and anti-inflammatory effects. Studies from large populations, including data from the blue zones, regions where people consistently live past 100, show that diets high in plants are associated with lower risks of cardiovascular disease, type 2 diabetes, and even certain cancers. One of the key mechanisms here is inflammation. Chronic low-grade inflammation is a common denominator in nearly every modern disease, and plant-based diets tend to be anti-inflammatory because they're high in fiber, antioxidants, and lower in saturated fats. Now let's contrast that with meat-based diets. Meat-based or animal-based diets, especially when done in a controlled, intentional way, can be nutrient-dense. They provide complete proteins, vitamin B12, heme iron, zinc, creatine, carnosine, and other compounds that are either absent or poorly absorbed from plant foods. When people shift toward carnivore or ketogenic styles of eating that emphasize animal products, some report reductions in autoimmune symptoms, weight loss, improved insulin sensitivity, and mental clarity. This isn't anecdotal. Research into low-carb and carnivore-style diets is showing promise in treating metabolic syndrome, obesity, and even certain neurodegenerative conditions. But here's the nuance. The health effects of a meat-based diet depend heavily on which meats and how they're prepared. Ultra-processed meats, bacon, hot dogs, sausages, are associated with increased risks of colorectal cancer and cardiovascular disease. But grass-fed beef, pasture-raised eggs, wild-caught fish, these are entirely different nutritional profiles. They contain omega-3 fats, conjugated linoleic acid, and fat-soluble vitamins like A, E, and K2, which can be highly beneficial. And on the flip side, not all plant-based diets are healthy either. A diet centered on refined grains, sugary smoothies, seed oils, and fake meat alternatives can still be technically plant-based, but drive metabolic dysfunction, obesity, and inflammation. This is why context matters. Your metabolic state, your genetic predisposition, your gut microbiome, your lifestyle, they all affect how you respond to a particular diet. Someone with insulin resistance might thrive on a low-carb, meat-based regimen, while someone else might feel their best on a plant-forward Mediterranean pattern. There's also the gut microbiome to consider. Fiber from plant foods feeds beneficial bacteria in the colon that produce short-chain fatty acids like butyrate, compounds that regulate inflammation, strengthen the gut barrier, and even influence mood and cognition. But animal foods aren't void of microbiome impact either. Certain amino acids and fats from meat can feed different microbial strains that might be helpful in other ways, such as supporting bile metabolism or producing neuroactive compounds. So instead of asking, which diet is better, maybe we should ask, which diet is better for this person, in this context, at this time in their life? Nutrition is not a religion. It's not a battle between plants and animals. It's a tool. And just like any tool, it should be applied with precision, with understanding, and with flexibility. If you're healing from leaky gut, autoimmune conditions, or long-term inflammation, temporarily going toward a meat-based elimination diet might be therapeutic. If you're aiming to prevent cardiovascular disease, improve your microbiome diversity, or reverse metabolic syndrome, then a whole food plant-based approach may be more effective. And the goal is never just weight loss or symptom suppression. The goal is resilience, the ability of your body to adapt, to repair, to respond appropriately to stress. Whether that stress is dietary, emotional, infectious, or environmental. Here's something else that gets lost in the debating: lifestyle synergy. Diet doesn't work in isolation. Regular exercise, quality sleep, sunlight exposure, breathwork, and social connection. These are all equally powerful levers for health. In fact, when you combine a moderate diet, plant-rich or meat-rich, with walking, fasting, and good sleep, the effects multiply. In conclusion, the best diet isn't about loyalty to one camp. It's about listening to your body, looking at the labs, checking your inflammation markers, your insulin sensitivity, your gut symptoms, and then adjusting accordingly. It's about combining ancient wisdom with modern science, and realizing that human health is complex, dynamic, and beautifully adaptable. So whether you lean toward the garden or the pasture, let it be in service of the same mission. A body that thrives, a mind that's clear, and a life that's full of energy, purpose, and longevity. Every diet sends biochemical signals that influence inflammation, immunity, and longevity. When we consume food, we're not merely fueling our bodies with calories or satisfying hunger. We're engaging in a complex biochemical conversation between our environment and our cells. Each bite of food delivers instructions to the body, and those instructions can either upregulate or downregulate gene expression, influence hormone levels, activate or suppress immune responses, and impact the delicate balance between oxidation and antioxidant defense. This biochemical signaling is continuous and accumulative. It means that the food we choose regularly, day after day, shapes the long-term trajectory of our health far more than we often realize. The types of macronutrients we consume, proteins, fats, and carbohydrates, all have unique signaling roles. Proteins from animal sources, for example, contain all essential amino acids and tend to stimulate the pathway more significantly than plant proteins. While mTOR is important for growth and repair, chronic overstimulation of this pathway has been linked to accelerated aging and increased risk of cancer. On the other hand, plant-based diets, which often deliver lower levels of methionine and leucine, may promote longevity by promoting a mild inhibition of this pathway, mimicking some of the effects of caloric restriction. Beyond macronutrients, micronutrients and phytochemicals found in whole foods act as signaling agents. Polyphenols in berries, sulforaphane in broccoli sprouts, curcumin in turmeric, all of these compounds interact with cellular signaling pathways. They can activate phase 2 detoxification enzymes, reduce oxidative stress, and modulate inflammatory cascades. For example, polyphenols activate the NRF2 pathway, which boosts the production of endogenous antioxidants like glutathione. This reduces cellular damage over time and may play a protective role in age-related diseases. The food we consume also influences the immune system in powerful ways. A diet high in refined sugars and processed fats tends to promote systemic inflammation by increasing the production of pro-inflammatory cytokines like TNF-alpha and interleukin-6. This chronic low-grade inflammation weakens immune surveillance and contributes to the pathogenesis of many modern diseases, including heart disease, type 2 diabetes, and even Alzheimer's. Conversely, a diet rich in anti-inflammatory foods like leafy greens, fatty fish, nuts, and colorful vegetables supports immune modulation. The omega-3 fatty acids from cold water, for instance, are converted into specialized pro-resolving mediators, SPMZs, which not only reduce inflammation but help actively resolve it. Another key layer of biochemical signaling occurs through the gut microbiome. The food we eat acts as either a prebiotic or a microbial modulator. High-fiber diets, rich in resistant starches and plant fibers, encourage the growth of beneficial microbes like Bacteria and Akkermansia. These microbes ferment fiber into short-chain fatty acids, such as butyrate, which serve as fuel for colon cells, enhance the integrity of the gut barrier, and reduce systemic inflammation. In contrast, diets low in fiber and high in saturated or processed meats can shift the microbial balance toward more pathogenic strains that produce endotoxins and drive inflammation across the gut-brain and gut-liver axes. The hormonal environment is also directly impacted by dietary choices: insulin, cortisol, leptin, ghrelin, thyroid hormones. Each of these is influenced by macronutrient ratios and meal timing. High-carbohydrate meals rich in refined sugars spike insulin and can lead to insulin resistance over time, a condition that underpins metabolic syndrome. Meanwhile, diets higher in healthy fats and proteins may blunt this response and improve insulin sensitivity. Even our circadian rhythm and sleep quality are influenced by what and when we eat, which in turn feeds back into the hormonal and immune systems. Through these various layers, genetic expression, inflammatory pathways, microbiome composition, hormonal balance, and mitochondrial function, our dietary patterns shape the aging process and our resistance to disease. What we eat regularly becomes a blueprint for how our body adapts, heals, and functions at every level. Whole food plant-based diets support cardiovascular and metabolic health through fiber, antioxidants, and polyphenols. These dietary components work together in a synergistic way to influence nearly every major system of the body, especially the cardiovascular system and metabolic processes such as blood sugar regulation, insulin sensitivity, lipid metabolism, and systemic inflammation. When plant-based diets are structured around whole, minimally processed foods, like leafy greens, legumes, berries, nuts, seeds, whole grains, and cruciferous vegetables, they deliver an incredibly dense array of nutrients that act at the cellular and molecular level to optimize health and reduce disease risk. Fiber is one of the foundational elements of plant-based nutrition that plays a direct and measurable role in cardiovascular and metabolic outcomes. Soluble fiber, found in foods like oats, beans, apples, and flax seeds, binds to bile acids in the intestine and helps to reduce cholesterol levels by facilitating the excretion of cholesterol from the body. This can lower LDL cholesterol, sometimes referred to as bad cholesterol, and has been shown in clinical trials to reduce the risk of coronary artery disease. Insoluble fiber, while not absorbed, contributes to satiety, regulates bowel movements, and supports detoxification by ensuring proper transit time of waste materials. High-fiber diets also blunt the postprandial glucose response, meaning that after meals, blood sugar levels rise more gradually. This is essential in reducing the risk of insulin resistance, which is a precursor to type diabetes and a driver of many metabolic disorders. Beyond fiber, plant foods are rich in antioxidants, natural compounds that neutralize free radicals and reduce oxidative stress. Oxidative stress is a key contributor to progression of atherosclerosis, the buildup of plaques in arteries that can lead to heart attacks and strokes. Antioxidants like vitamin C, vitamin E, selenium, and numerous polyphenolic compounds found in plants actively scavenge reoxygen species and inhibit the oxidation of LDL cholesterol, a critical step in the development of arterial plaques. Green leafy vegetables like spinach and kale, for example, are not only high in fiber, but are also rich sources of lutein and folate, both of which have been associated with lower homocysteine levels and improved endothelial function. Berries such as blueberries and raspberries contain anthocyanins, which are powerful antioxidants that have been shown to improve vascular elasticity and reduce systolic blood pressure. Polyphenols, which are a large group of plant-derived phytochemicals, exert particularly interesting effects on cardiovascular and metabolic health. Found in colorful fruits and vegetables, tea, coffee, herbs, spices, and dark chocolate, polyphenols have been studied for their role in modulating key enzymes and pathways that affect inflammation, insulin sensitivity, blood pressure, and lipid metabolism. For instance, flavonoids found in citrus fruits and onions have been shown to inhibit the angiotensin-converting enzyme, ACE, leading to vasodilation and blood pressure reduction. Resveratrol, a polyphenol found in grapes and peanuts, may activate sirtuins, a family of proteins linked to longevity and improved metabolic efficiency. Curcumin, found in turmeric, has demonstrated the ability to reduce inflammatory markers like C-reactive proteins CRP and interleukin-6, both of which are associated with cardiovascular risk. Plant-based diets are also naturally low in saturated fats and cholesterol, both of which have been implicated in the development of cardiovascular disease when consumed in excess. By replacing saturated fats with polyunsaturated and monounsaturated fats from sources like avocados, olives, flax seeds, and walnuts, plant-based diets can help improve lipid profiles, reduce triglyceride levels, and support overall heart health. Additionally, the phytosterols found in nuts and seeds structurally resemble cholesterol and compete for absorption in the gut, which can further contribute to reductions in circulating cholesterol levels. In terms of metabolic regulation, Whole food plant-based diets help to stabilize blood sugar by providing a slow and steady release of glucose due to their high fiber content and low glycemic index. Legumes, whole grains, and non-starchy vegetables contribute to a more balanced glucose curve after meals and reduce the risk of insulin spikes. Over time, this promotes improved insulin sensitivity, reduces the risk of metabolic syndrome, and supports weight loss or weight maintenance. Obesity is one of the strongest predictors of insulin resistance, and plant diets are associated with lower body mass index (BMI) across multiple population studies. Another important aspect is the role of plant-based diets in reducing chronic inflammation, which underlies many chronic diseases, including cardiovascular and metabolic disorders. Foods such as tomatoes, leafy greens, berries, and nuts contain anti-inflammatory compounds that interfere with proliferatory cytokines and reduce the activity of nuclear factor kappa B NFJ, a key transcription factor involved in the inflammatory response. Chronic low-grade inflammation not only damages blood vessels and impairs insulin signaling, but also accelerates biological aging and increases vulnerability to degenerative diseases. Plant-based diets also contribute to improved endothelial function, the health of the inner lining of blood vessels. Nitric oxide, a molecule essential for blood vessel dilation and overall vascular tone, is upregulated by dietary nitrates found in beets, arugula, and spinach. Enhanced nitric oxide production leads to improved circulation, lower blood pressure, and reduced risk of cardiovascular events. In patients with established heart disease, switching to a plant-based diet has even demonstrated reversal of atherosclerotic plaque in some clinical trials, showing not only preventive but potentially regenerative effects on vascular health. The gut microbiome also plays a central role in the health benefits of plant-based eating. The fibers and polyphenols found in plants act as prebiotics, selectively feeding beneficial bacterial strains that produce short-chain fatty acids like butyrate. These fatty acids are anti-inflammatory, strengthen the gut barrier, and improve metabolic signaling throughout the body. A diverse and robust microbiome contributes to improved glucose tolerance, reduced appetite dysregulation, and better lipid metabolism. Emerging research continues to show that plant-rich diets foster environments that support metabolic homeostasis and protect against obesity and related diseases. Meat-based diets provide bioavailable nutrients and may benefit certain individuals with autoimmune or metabolic issues. Animal-sourced foods deliver complete proteins with optimal ratios of essential amino acids, particularly leucine, which robustly stimulates muscle protein synthesis and supports preservation of lean mass during weight loss or aging. They supply vitamins and minerals in forms that are readily absorbed, vitamin B12 exclusively, heme iron with higher absorption efficiency than non-heme iron, performed vitamin A retinol that bypasses the variable conversion of carotenoids, zinc without the phytate inhibitors common in grains and legumes, vitamin K2 as MK4 in egg yolks and liver, and long-chain omega-3 fatty acids EPA and DHA from fatty fish that do not rely on the limited alpha-linolenic acid conversion pathway. Carnitine, creatinine, carnosine, and anserine, conditionally essential or physiologically supportive compounds, occur naturally in meat and can influence mitochondrial function, buffering capacity, neurocognitive performance, and glycemic control in ways that are difficult to replicate with plant-only diets. For people with autoimmune conditions or severe gastrointestinal sensitivity, a carefully structured meat-forward or even temporary elimination style animal-based diet can reduce exposure to potential plant-derived triggers such as lectins, oxalates, certain FODMAPs, and alkaloids. By narrowing antigenic load and simplifying the food matrix, some individuals report reductions in symptoms like bloating, joint pain, skin rashes, and brain fog, possibly through decreased intestinal permeability, lower immune activation, and reduced fermentative gas production in the gut. In clinical practice and emerging literature on low residue or fiber diets for active inflammatory bowel disease flares, strategic reduction of fermentable substrates can sometimes provide symptomatic relief while the intestinal mucosa heals, after which a gradual reintroduction of tolerated fibers and polyphenols can be attempted to restore microbial diversity and butyrate production. And from a metabolic standpoint, Higher protein, lower carbohydrate meat-based patterns often yield improvements in glycemic variability, fasting insulin, HOMA-IR, triglycerides, and the triglyceride to HDL ratio, particularly in insulin-resistant or type 2 diabetic populations. Lower carbohydrate intake reduces the immediate need for insulin secretion, decreasing postprandial glucose excursions and facilitating lipolysis. Protein's thermic effect increases post-meal energy expenditure, while its satiety effect, mediated through hormones like GP1, PI1, and CK, can spontaneously reduce caloric intake without conscious restriction. Individuals with severe hyperinsulinemia frequently find it easier to a diet that minimizes foods most rapidly driving insulin demand. And for some, a structured meat-forward pattern is the most straightforward execution of that principle. Nutrient density and bioavailability, however, do not automatically translate into broad-spectrum health benefits without attention to quality and context. The differential effects between minimally processed, pasture-raised ruminants, wild-caught fish, and eggs, processed meats high in nitrates, advanced glycation end products, AGEs, and industrial seed oils are profound. High temperature, dry heat cooking methods can increase heterocyclic amines and polycyclic aromatic hydrocarbons pH, compounds linked to DNA damage. Slower, lower temperature techniques, brief searing followed by oven finishing, sous vide, or marinating meats with antioxidant-rich herbs and acids can mitigate these byproducts. Including oily fish such as sardines, mackerel, and salmon ensures a favorable EPA-DHA intake to balance arachidonic acid-derived eicosanoids and to support resolution of inflammation through specialized pro-resolving mediators. There are trade-offs to consider. Ultra-low fiber versions of meat-based diets can reduce microbial diversity and short-chain fatty acid production, potentially impacting gut barrier integrity and systemic inflammation over time for some individuals. LDL-C in a palm can rise substantially in a subset of people. Often lean, insulin-sensitive, hyperresponders, necessitating careful monitoring of LDLP, lipoprotein OHCRP, and oxidized LDL, along with contextual markers like CAC scores when appropriate. The absence of certain plant-derived polyphenols that ACT2 pathway or inhibit NFJB could diminish hormetic antioxidant signaling, unless compensated by other lifestyle hormetic stressors, such as exercise, cold exposure heat therapy, or intermittent fasting. Extremely restrictive carnivore implementations may also risk inadequate of folate, vitamin C, manganese, and magnesium unless organ meats, shellfish, bone broths, and mineral-rich waters are incorporated, or unless targeted supplementation fills those gaps. Electrolyte management becomes crucial when a meat-based diet is also ketogenic, as natriuresis of fasting and low insulin states increases renal sodium loss, which can secondarily deplete potassium and magnesium, contributing to fatigue, cramps, and palpitations if not proactively addressed. Individuals with gallbladder insufficiency or pancreatic exocrine insufficiency may initially struggle with the fat load of certain meat-centric approaches, requiring digestive support or a phased macronutrient transition. People with hereditary iron overload, hemochromatosis, or high ferritin may need to limit heme iron intake or donate blood regularly, highlighting again that personalization and lab-guided iteration are more important than allegiance to any dietary ideology. A pragmatic strategy for those exploring meat-based nutrition therapeutically is to define a time-bound elimination window, aggressively track symptoms, biomarkers, and body composition, and then strategically reintroduce low-antigen, high-benefit plant foods, like berries, avocados, olives, fermented vegetables, extra virgin olive oil, herbs, and spices, to regain microbiome diversity and phytochemical hormesis while keeping symptom triggers out. Others may elect to remain predominantly animal-based but include fiber sources such as partially hydrolyzed gar gum or psyllium to support bowel regularity and butyrate production without reactivating prior intolerances. Throughout, frequent monitoring of metabolic health markers, fasting glucose, baric, insulin, triglycerides, HDL, cardiovascular risk markers, APOB, LDLP, ELEPSA, SCRP, and micronutrient status ensures that the theoretical advantages of bioavailability translate into real-world, long-term health gains rather than transient improvements obscuring emerging risks. In short, meat-based diets can be potent clinical tools when deployed with precision, nutrient density, and an understanding of individual biochemistry, but they work best when integrated into an adaptive framework that respects metabolic individuality, continually measures objective outcomes, and remains open to iterative reintroduction of non-triggering, health-promoting plant compounds as tolerance of the quality and processing of both plant and animal foods are more important than the category itself. Whether a food is plant-based or animal-based tells us very little about its health impact without understanding how it was grown, raised, processed, prepared, and combined with other dietary components. Whole forms of both plants and animals offer bioavailable nutrients, but when those same foods are processed, stripped of fiber or essential compounds, exposed to heat-induced toxins, or blended with industrial ingredients, their health effects can shift dramatically. This is a key reason why many dietary studies show conflicting outcomes, because they often fail to differentiate between ultra-processed and whole food versions of the same basic food category. And take meat, for example. There is a clear distinction between a grass-fed cooked gently at home and a highly processed sausage made with preservatives, fillers, seed oils, and artificial flavors. The former provides complete proteins, heme iron, B vitamins, and healthy fats without excessive additives. The latter may still contain protein but introduces nitrate phosphates, advanced glycation end products, and oxidized fats that have been linked to increased risks of colorectal cancer and cardiovascular disease. The way the meat is cooked matters as well. High-temperature methods like frying or grilling from heterosiliceamines and polycyclic aromatic hydrocarbons, especially when meats are charred or overcooked. These compounds have been associated with DNA damage and increased cancer risk in animal models, although their direct impact in humans depends on other dietary and lifestyle factors. Conversely, slow cooking, braising, or sous vide methods reduce the formation of these harmful compounds and preserve more of the meat's nutrients. Similarly, not all plant-based foods are beneficial simply because they come from a plant. Many modern plant-based products are highly processed, engineered to mimic meat or dairy, and loaded with additives, stabilizers, emulsifiers, and inflammatory seed oils. Ultra-processed plant-based foods often include refined grains, high-fructose corn syrup, artificial flavorings, and synthetic vitamins. These can drive inflammation, elevate blood glucose, disrupt the gut microbiome, and promote weight gain. A bowl of lentils, vegetables, and olive oil has a profoundly different metabolic and inflammatory effect than a plate of plant-based nuggets made from isolated soy protein, wheat gluten, and canola oil. One delivers fiber, polyphenols, and prebiotic compounds that support gut health, while the other may contribute to metabolic dysfunction and oxidative stress despite its plant origin. The source and growing method of plant foods also matter significantly. Leafy greens and berries grown in nutrient-rich soil without pesticides are not the same as their conventionally grown counterparts treated with glyphosate and synthetic fertilizers. Pesticide residues, heavy metals, and environmental toxins can accumulate in plant foods and influence health outcomes over time. Additionally, monocropping and soil depletion reduce the mineral content of many modern fruits and vegetables. This means that eating whole plant foods from sustainable, organic, or regenerative sources can provide more micronutrients and fewer toxicants compared to conventionally farmed versions. The same is true for animal products. Animals raised on pasture, with access to natural diets and sunlight, produce meat, milk, and eggs with higher omega-3 fatty acids, more conjugated linoleic acid, better vitamin D levels, and fewer inflammatory compounds than animals raised in confinement, fed corn and soy, and treated with antibiotics and hormones, even within whole food forms. How foods are combined and consumed plays a role in their health impact. Adding extra virgin olive oil to steamed vegetables enhances absorption of fat-soluble vitamins and antioxidants like beta-carotene and lutein. Including vinegar or lemon juice with meals can lower the glycemic impact of carbohydrate-rich plant foods. Eating fiber-rich vegetables before starchy or sugary items can reduce post-meal blood sugar spikes. Conversely, combining refined carbs with processed meats, such as white bread and deli meats, may synergistically increase inflammatory markers and cardiovascular risk. The broader dietary context determines whether a given food exerts a healing or harmful effect. A small portion of high-quality cheese in a Mediterranean-style diet may be beneficial, while the same cheese consumed daily alongside fast food, soda, and low-fiber foods may contribute to disease. The conversation about food quality must also include additives, preservatives, and packaging materials that are common in both plant-based and meat-based processed foods. Artificial sweeteners, emulsifiers like polysorbate 80, and preservatives such as sodium benzoate have been shown in some studies to disrupt gut bacteria, increase intestinal permeability, and drive low-grade inflammation. Plastics and bisphenols used in packaging can leach into food and interfere with hormone signaling. These exposures, though often considered minor in isolation, can compound over time and contribute to metabolic disruption and chronic disease. Choosing fresh, unpackaged, or minimally processed foods helps to minimize these cumulative exposures and preserve the nutritional integrity of the diet. Another critical aspect of food quality is the preservation of structure and fiber. In whole fruits, vegetables, legumes, and grains, the cell wall structure slows digestion and creates a gradual release of glucose into the bloodstream. Processing methods that break down this structure, juicing, puffing, extruding, and refining, eliminate this natural benefit and can convert a once healthy food into one with a glycemic index similar to that of sugar. For example, eating whole oats provides beta-glucans and stable blood sugar control, while instant oats may spike insulin levels more dramatically. Similarly, a whole apple includes pectin and polyphenols that support satiety and gut health, while apple juice lacks fiber and delivers a concentrated dose of fructose without its protective matrix. In animal foods, preserving the integrity of organs and connective tissues can enhance nutritional density. Nose-to-tail eating practices, which include organ meats, bone broths, and collagen-rich cuts, provide a wider array of nutrients than muscle meat alone. Liver is a powerhouse of B12, vitamin A, copper, and choline. Bone broths deliver glycine and gelatin that support joint and gut health. When only lean muscle meat is consumed, especially in the absence of balancing nutrients from plants or organs, certain imbalances can arise, such as methionine excess without glycine, which may influence malation and longevity pathways. Traditional dietary patterns that honored whole animal utilization may have had hidden advantages in balancing these micronutrient ratios naturally. Ultimately, the fixation on food categories, plant versus animal, can distract from a more meaningful distinction: processed versus whole, industrial versus traditional, inflammatory versus anti-inflammatory. A diet filled with quality, thoughtfully sourced, minimally processed foods, whether plant-based, animal-based, or a combination, can support optimal health when aligned with the individual's needs, preferences, and metabolic conditions. Prioritizing quality over ideology allows for more flexibility, better adherence, and more nuanced health outcomes in real-world settings where perfection is rarely achievable and balance is more sustainable. Gut microbiome responses vary based on fiber, fat, and amino acid intake from both diet types. The human microbiome is an intricate and dynamic ecosystem composed of trillions of microbes that communicate constantly with the immune system, nervous system, and metabolic pathways. The foods we eat shape the diversity, resilience, and function of this microbial community, and the differences in dietary inputs from plant-based and meat diets result in distinct microbial profiles with unique metabolic outputs. The fiber-rich nature of whole plant foods provides the primary fuel for beneficial bacteria that produce short-chain fatty acids, such as butyrate, acetate, and propionate. These metabolites play key roles in maintaining integrity of the intestinal barrier, modulating immune responses, and influencing gene expression in cells lining the colon. When individuals consume a plant-based diet high in legumes, vegetables, fruits, and whole grains, the microbiome tends to increase in diversity, particularly in species like bifidobacteria and lactobacilli. These bacteria thrive on fermentable fibers and resistant starches, generating metabolites that reduce inflammation, regulate appetite hormones, and contribute to mucosal healing. Butyrate, in particular, supports colonic epithelial cells, helps regulate T regulatory cell populations, and dampen overactive immune responses that are involved in inflammatory bowel disease, autoimmune conditions, and allergies. Fiber also increases stool bulk and transit time, which aids detoxification helps bind toxins, estrogens, and bile acids for excretion. Diets that consistently include diverse plant fibers tend to correlate with better gut barrier integrity, less endotoxin leakage into the bloodstream, and lower systemic inflammation markers like C-reactive protein and interleukin-6. Polyphenols from plant foods also play a significant role in shaping the microbiome. These compounds are not fully absorbed in the small intestine, and thus reach the colon, where they act as prebiotics and modulators of microbial populations. Polyphenol-rich foods such as berries, cocoa, green tea, and herbs influence the balance of microbial species and stimulate beneficial bacteria to increase their production of anti-inflammatory metabolites. Some microbes can even biotransform polyphenols into more bioactive compounds that further exert antioxidant and anti-proliferative effects within the gut and beyond. This bidirectional relationship between polyphenols and gut microbes is part of the reason why diets like the Mediterranean diet show such strong links to improved gut and metabolic health, and in contrast, meat-based diets often result in reduced microbial diversity and a shift toward different dominant bacterial species, especially if the diet lacks sufficient plant fiber. Certain amino acids and proteins from meat are fermented gut bacteria to metabolites like putrescine, cadaverin, hydrogen sulfide, and branched-chain fatty acids. While some of these byproducts can support intestinal function, when present in small amounts, high levels, particularly in the context of low fiber, may contribute to increased colonic pH, gut barrier disruption, and inflammation. Red meat consumption, especially when high and unbalanced by fiber, has been associated with increased production of trimethylamine (TMA), which is oxidized in the liver to TMAO. Elevated TMO levels have been correlated with cardiovascular risk in some observational studies, although the causality and context remain debated. Individuals with a fiber-rich background diet or diverse microbiome may produce less TMO from the same meat intake than those with low-fiber diets, showing how microbiome resilience can buffer potential risks. Despite these concerns, certain elements of meat-based diets can positively influence the microbiome, particularly when organ meats, collagen-rich cuts, fermented animal products, and bone broths are included. Collagen and glycine provide substrates for gut mucosal repair, while fermented dairy like kefir and yogurt introduce live bacterial strains, such as lactobacillus and bifidobacterium, that may help restore balance in disrupted microbiomes. Bone broth supplies amino acids and minerals that support gut barrier repair and can be especially useful in low-residue elimination diets used in inflammatory gut conditions. In some individuals with small intestinal bacterial overgrowth, SBIBS, or autoimmune disease, temporary reduction of fermentable carbohydrates and increased intake of meat-based nutrition can lead to symptom relief and reduced bloating, highlighting that fiber is not universally beneficial in all cases. Particularly when the microbiome is imbalanced or compromised. Dietary fat also plays a major role in microbial shifts. High intake of saturated fats from processed meats and dairy has been associated with an increase in bile-tolerant species, such as Bilophila wadsworthia, which may drive inflammation in susceptible individuals. These species thrive when bile acids are secreted in high quantities, which occurs with high-fat meals. On the other hand, monounsaturated and omega-3 polyunsaturated fats from sources like fish, nuts, avocados, and olive oil have been shown to foster beneficial shifts in microbial composition and improve gut barrier function. Fish oils can reduce levels of lipopolysaccharide-producing bacteria, lower intestinal permeability, and modulate immune responses within the gut-associated lymphoid tissue. The effect of dietary protein on the microbiome is also influenced by its source, preparation, and balance with other nutrients. While protein is essential for health, excessive protein not buffered by fermentable carbohydrates may result in putrefactive fermentation, a process that generates ammonia and phenolic compounds that can be harmful in excess. This does not mean that meat is inherently damaging, but rather that high-protein, low-fiber diets can create an imbalance in fermentation patterns, including protective elements such as gelatin, organ meats, herbs, and polyphenols can help mitigate some of these effects. Furthermore, individuals with robust digestive capacity, healthy bile production, and low levels of gut inflammation may tolerate higher protein loads more effectively than those with compromised digestion. The timing and frequency of meals, meal composition, and overall energy intake all interact with microbiome rhythms. Both plant-based and meat-based diets can support microbial health when timed to align with circadian rhythms. When periods of digestive rest are built into the day, and when nutrient density is prioritized over quantity. Fasting periods, such as intermittent fasting or time-restricted eating, have been shown to increase microbial diversity and reduce markers of metabolic disease. These benefits can be achieved with either diet type, provided the meals consumed are whole, unprocessed, and rich in supportive compounds. Microbiome diversity can also be influenced by lifestyle factors such as stress, sleep, exercise, and environmental exposures, showing that the gut's response to diet is part of a larger ecosystem of inputs that must be considered together in clinical settings. Stool testing, metabolomic profiles, and symptom tracking are being used more frequently to determine how a person's unique microbiome responds to specific dietary interventions. Some individuals flourish on high-fiber, polyphenol-rich diets, while others do better initially with simplified, lower-fiber, higher-protein approaches to calm immune overactivation and digestive symptoms. Over time, microbiome diversity is a shared goal in most health strategies, and it can be achieved through many dietary paths depending on the individual's current health state, genetics, and microbiome baseline. The best diet is personalized, context-driven, and works in synergy with lifestyle factors like sleep, exercise, and stress. Nutrition does not exist in a vacuum and cannot be properly evaluated or implemented without considering the broader physiological, psychological, and environmental circumstances of an individual. One person may thrive on a high-fiber, plant-centric diet due to a healthy microbiome, low stress, and excellent digestive function, while another may require a meat-focused, low-residue approach because of autoimmune flare-ups, gut permeability issues, or unresolved metabolic dysfunction. The personalization of diet involves acknowledging that genetic variability, microbiome composition, food sensitivities, metabolic flexibility, and even personality traits and cultural habits all influence dietary tolerance and long-term sustainability. Metabolic individuality is one of the most overlooked aspects of dietary decision-making. While two people may eat the same meal, their glycemic responses, lipid metabolism, and hormonal cascades can be entirely different. Continuous glucose monitoring studies have revealed that some individuals experience large blood sugar spikes after eating bananas or oats, while others maintain flat glucose curves. Similarly, the degree to which saturated fats raise LDL cholesterol varies dramatically from person to person, influenced by genetics, gut microbiota, and overall dietary pattern. Some individuals are lean mass hyper-responders who see a sharp rise in LC on low-carb, high-fat diets, while others experience normalization of lipids and inflammation. Personalization means these differences are not pathologized but understood within the context of total health markers, including ApoB, HDL, triglycerides, insulin levels, inflammation markers, and body composition, and context matters deeply when evaluating a person's dietary needs. Someone with insulin resistance, elevated triglycerides, and non-alcoholic fatty liver disease may respond more favorably to a lower carbohydrate, protein-forward diet to reduce hepatic fat, normalize fasting insulin, and stabilize energy levels. In contrast, an individual with elevated homocysteine atherosclerotic burden and a family history of cardiovascular disease may benefit more from a plant-forward diet that emphasizes antioxidants, nitric oxide precursors, and fiber-rich legumes. A woman in early perimenopause may require a different balance of protein, fats, and phytoestrogens compared to a young male athlete recovering from intensive strength training. The idea that one diet is optimal for all disregards the phases of life, sex-specific hormone profiles, and the evolving nature of individual physiology. Lifestyle factors interact with diet at every level. A diet that looks perfect on paper may fail to deliver results if the individual is sleep-deprived, chronically stressed, or sedentary. Poor sleep quality elevates cortisol, disrupts hunger hormones like ghrelin and leptin, increases insulin resistance, and promotes cravings for calorie-dense, nutrient-poor foods. Stress activates the sympathetic nervous system and shifts digestive priorities away from optimal enzymatic function and nutrient absorption. Someone under chronic psychological or physical stress may not tolerate raw vegetables, legumes, or high-fiber meals well, leading to bloating, cramping, or nutrient deficiencies despite eating what is considered a healthy diet. Exercise also modifies dietary needs and nutrient turnover. A physically active person may require more calories, electrolytes, protein, and antioxidants, while a sedentary individual may struggle with blood sugar regulation and would benefit from reduced portion sizes and lower glycemic loads. The timing and rhythm of eating also play a significant role in how diet impacts health. Intermittent fasting, time-restricted eating, and circadian-aligned feeding can all enhance metabolic flexibility, improve insulin sensitivity, and support digestive efficiency. For some individuals, compressing the eating window to 8–10 hours during daylight hours can reduce nighttime glucose spikes, lower inflammation, and improve sleep quality. For others, particularly those with adrenal dysregulation or hypothyroidism, excessively long fasting windows may cause fatigue, mood instability, or poor thyroid conversion. Meal timing, spacing, and consistency must therefore be tailored to individual stress levels, work schedule, activity level, and hormonal rhythm. Cultural, emotional, and psychological relationships with food also influence dietary sustainability and success. A diet may be nutrient-rich and anti-inflammatory, but if it feels socially isolating, emotionally unsatisfying, or logistically impractical, adherence will suffer. Food is not just fuel, it's community, memory, comfort, and ritual. Personalization includes aligning dietary choices with cultural values, food availability, cooking skills, and budget. For some, preparing plant-rich meals is a grounding ritual. For others, sourcing regenerative meats and using ancestral cooking methods connects them to heritage and wellness. In either case, sustainability and enjoyment are as crucial as nutrient density and macronutrient balance. Our advanced testing and biomarker tracking are becoming increasingly accessible and can guide dietary personalization with more precision. Tools like continuous glucose monitors, lipid particle testing, hormone panels, food sensitivity testing, microbiome sequencing, and metabolomics can help identify how the body is responding to specific foods and meal structures. These insights allow individuals to move beyond guesswork and to targeted strategies that address root causes rather than symptoms. For example, if a person discovers high zonulin or lipopolysaccharide levels, they may opt for a gut repair protocol that includes glutamine, collagen, and low FODMAP meals. If another person shows low omega-3 index or elevated CRP, their focus may shift toward fatty fish, flaxseed, and anti-inflammatory herbs. A personalization also includes periodic reassessment. A diet that works well during fat loss may not be optimal for maintenance, muscle growth, or recovery from illness. As life circumstances change, so do dietary needs. Hormonal shifts, aging, physical injuries, changes in sleep or work schedules. All these factors affect nutritional requirements. People recovering from chronic illness may need higher levels of protein, micronutrients, and mitochondrial support. Athletes in peak training may require additional antioxidants and electrolytes. During periods of low activity or high mental stress, lighter meals and more adaptogenic compounds may be preferable. Health is not a fixed target but a constantly moving balance point that requires ongoing attention and adaptability. A truly effective dietary approach is one that supports the body's changing needs while being flexible enough to account for real-world constraints and preferences. It integrates data with intuition, science with culture, and biology with behavior. It considers not just what is eaten, but when, why, and how it is eaten. It recognizes that people are metabolically, emotionally, and environmentally different, and that those differences are not flaws to be corrected but variables to be respected and worked with. Personalization is not complexity for its own sake. It is the necessary path to genuine, long-lasting health outcomes in a world of ever-changing inputs and individual uniqueness.